As a grower, you are always working towards achieving the best quality of your crop. To make this possible, Eurofins Agro provides you all essential information related to water, fertilization, growing media, and plant health. In this webinar, Jan Hardeman explains the importance of testing of water quality and microorganisms in relation to the efficacy of the disinfector.
Hello, my name is Jan Hardeman. In this presentation, I'd like to tell you more about water quality and microorganisms in a greenhouse. First, I'd like to tell you more about water quality. What do we define with water quality? And I'd like to tell you more about the methods to analyze microorganisms. And where do we find the microorganisms in the greenhouse? What do we detect in the samples we receive in the laboratory? Now, the first, I'd like to tell you more about water quality. It's the pH, of course, EC level. It's analyzed a lot in the greenhouse to check the levels, to grow optimal crops. Of course, the nutrients are very important. Sodium and chloride, especially if you recirculate the water, sodium and chloride could be a problem if you have too high amounts in your water. The organic parts in the water, like organic fertilizers, biostimulants, the substrate gives also organic parts in the water. Chemical contamination, like accumulation of pesticides, heavy metals. There is a lot of contamination also in the water. And we can detect several of these compounds with specific techniques. And the focus in this presentation is about microorganisms. The plant pathogens, of course, very important to analyze, but also positive organisms are present in a greenhouse. Where can I find microorganisms in a greenhouse? There are several places in a greenhouse, but where are they? It's good to check where they are. They are hidden in specific places. It can be in a water source, A and B tank, fertigation system. There are a lot of tubes, irrigation systems in a greenhouse where these pathogens can hide. The crops, of course, there can be damage on the crop. The specific fungi and bacteria around the roots, they settle in the substrate around the roots. These fungi and bacteria or viruses can be detected also in the drain water and in the clean water tank. Is this tank really that clean, I expect? And are these fungi and bacteria pathogenic or not? If we look at the water treatments, there are several techniques in the greenhouse to treat the water, to disinfect the water, like ozone, UV light, heating systems, specific filtration techniques. Advanced oxidation techniques is a combination of UV and hydrogen peroxide, and specific disinfectants, like hydrogen peroxide, specific chlorine dioxide, and sodium hypochlorite. There are several methods, machines, disinfectants to disinfect the water. But are they working properly? Is the technique working fine? And there are several laboratory methods to detect the amount of fungi and bacteria in the water. And I'd like to tell you more about that. The laboratory methods. First, the plating techniques. These are specific techniques. We pour water on a growing media, the fungi and bacteria will grow, and this is a good indicator for the microbiological activity. We have also specific DNA techniques. It's Pcr techniques, it's specific techniques to detect the fungi and bacteria in the water. It's a fast measurement on species level, so very precise. Another method is gas chromatography. It's a specific technique. We call it our soil life monitor analysis. And in a later webinar, my colleague can tell you more about this specific method. But in this presentation, I like to focus on the first two, the plating techniques and the DNA techniques. If we look at the plating techniques, we have a specific disinfector check. So before and after a disinfector, we take a sample and we can detect the fungi and bacteria in the water. The micro check, on another place in the greenhouse, we can analyze the water, like water in water tanks, or the fertigation water you can send to the laboratory, or the A and B tank solution. Hygiene check is the same method in the laboratory, but you can send the swabs or wipes to the laboratory after you put them on a surface, and then you can send it to the lab for an analysis. For example, after a crop rotation. There are also species-specific media. We use for the disinfector check, micro check and hygiene check specific media where a lot of fungi and bacteria can grow, but there are also specific media where only a few species can grow, like Trichoderma, the good fungi. You see a picture. Trichoderma is forming nice colonies on a plate with a nice color, so it's easy to detect. The reporting is in colony forming units per milliliter water or colony forming units per swab. And we count the living bacteria and fungi in the water. If we look at the sampling of the disinfector check, it's very important to work sterile. Use specific gloves, disinfected tap, that's very important. If you have a metal tap, you can do it with heating, like you see on the picture, or you can use alcohol for a plastic tap. You can run the tap for a few minutes, and then you can sample it in a specific sterile bottle. It's very important to do it before and after disinfector, so you can get a good view of the working of the disinfector. You can send it to the laboratory with cooled transport. And what kind of parameters do we analyze within the disinfector check? First, the colony count. We pour water on a plate. The colonies are growing on the plate, so it are all microorganisms which are able to form colonies on this specific growing media. And the colony count is considered as an indicator, an hygiene parameter of the contamination of water. Also in the drink water industry, they use this specific colony count. Another parameter we analyze is the fungi. And we can analyze also the yeast on a specific growing media. The reporting is in the colony forming units per milliliter water. We made a few pictures of the laboratory. You see the colony count. On the left side, you see a plate with only a few colonies growing. So only a few colonies are present in the water. On the right side, you see a plate with a lot of dots. So a lot of colonies are growing. Fungi plate is a specific plate for the fungi. So only fungi will grow on this growing media. And you see on the left side only a few colonies, and on the right side a beautiful plate with a lot of dots. So a lot of fungi are growing. We count these plates, and you see a picture of it, how it works. So counting all the dots. And then we report the amount of colonies. First, the colony count, untreated in this example, more than 30,000 colonies growing on the plate. So that's quite a lot. Also total fungi, 9,000. It's a lot of colonies. The water is treated, for example, after the disinfector, and there are no colonies present. So this disinfector is working quite well. If you look at the targets, then we say the germination count, the colony count below 500 is okay for water after a disinfection technique. But every colony which is growing after disinfector is possible a bad one, so possible a plant pathogen. So if you look at the efficacy of disinfectors, what do we detect? In several samples, hundreds of samples we receive every year. On the horizontal axis you see the colony count, and on the vertical axis the percentage of the sample with that amount of colonies. Then you see the orange bar, you see the samples after disinfector in 2015, and the green column is 2020. So in samples after disinfector last year. And then you see a difference. 5 years ago, more than 50% of the disinfectors has shown more than 500 colonies in the water. In 2020, more than 20% above the target level. And you see it also, the low amounts of colonies, below 10 is last year almost 40%. 5 years ago, it was around 15%. So we can see that there is a focus on disinfectors. So they are working better and better. Because in the Netherlands, a lot of recirculation is now normal in the greenhouse, and within a few years all the water must be recirculated. Then it's important that your disinfector is working fine. So we see the machines are getting better and better, and more growers are checking the disinfectors or they are working fine or not. So after a disinfector, which are the possible causes of a high amount of colonies, so a high level of colony forming units? In the UV system, the low UV transmission value can be a cause. It's important that it is above the 75%. Bad maintenance, for example, old machines, old UV lamps. Wrong settings, the intensity of the UV, for example, in relation with the flow rate of the water. It's very important. And a possible cause is also the wrong sampling, the wrong place or not sterile. So there are a lot of causes, but you can check it with the analysis. Is your disinfector working fine or not? We go to another method, the DNA method with Pcr technique. It's a detection of more than 100 fungi and bacteria with this specific technique, and it's possible to do this in water, soil and substrate and plant material. It's fast and specific. Within 2 or 3 days you will receive the result in the email. It's highly sensitive and reliable, and it's an analysis on species levels. So we know which DNA is in which species. So it's then easy to detect quite a wide range of fungi and bacteria. If you look at the report of the DNA multiscan, then you can see the detection levels. So in this example, you see a lot of Fusarium, SPP, the subspecies, so it's the complete group of Fusarium, and a part of it is Fusarium oxysporum. The detection level is from 1 to 6. 1 will say it's a very low amount of DNA present. Detection level of 6, a very high amount of DNA present in the water or in the soil or in the crop. So after you receive the results, within a few days you can take the right measures. So what do we detect in water and growing media in hundreds of different samples? Then you see often Pythium is present. Also Fusarium and Phytophthora, very important primary pathogen which can cause a lot of damage in the crop. You see in 1/3 of the samples we will find it. But also the good organism Trichoderma is used a lot in greenhouses. you see it with the DNA test. Sometimes microorganisms are in unexpected places. In this example, we took the sample at the grower. You see Phytophthora and Pythium present in the fertilizer tank. You wouldn't expect this fungi growing in this highly concentrated tanks with that high amounts of EC. But they are growing there. This grower had quite a lot of problems with these fungi in his crop. So these fungi were growing in the fertilizer tanks. And also if we plate the water, we see a high amount of colonies growing on a plate. Fungi and also the colony count is quite high. Another analysis we offer is via our plant doctors. They have a lot of knowledge about plant pathogens, and it's a diagnostic service for diseased crops. And if you have a diseased crop, you like to know the primary cause, and our plant doctors will find them. They use a lot of different techniques. They can plate water, compost as substrates if you are looking for a specific fungi. They use several growing media where they grow the fungi and bacteria. And they use also other DNA techniques. They use all available techniques, including DNA sequencing techniques for unknown pathogens. So they are the specialists. They stop searching until they find the primary cause. And that's you want to know as a grower. You want to know what to do and which problem the cause is of your diseased crop. And if you look at the report, my colleagues, the plant doctors, they make pictures of the crop and sometimes also picture if they search it under the microscope. And they write a complete report by hand. You will receive it on the email with a description of the symptoms. What do they see when the material arrives in the laboratory? They give a description of the applied techniques and the detected fungi, bacteria, or other things they found in the crop, like deficiency problems. Maybe that's the cause of the problem. And at the end, they describe the primary cause. Often they find quite a range of other organisms, secondary organisms, but the primary cause you want to know, and they will find it. And at the end, they will give an advice what to do. So which analysis do you choose? DNA multiscan or plant doctor in plant material? If you have plant material with symptoms and you don't know the cause, you can send the plant material to our plant doctors. So send roots, stems and leaves. It's very important to send a complete crop because maybe the problem is in the roots and you will see it in the leaves. So the plant doctors like to receive the complete crop. But if you want to screen or monitor or confirm suspicion of one or more pathogens on the DNA multiscan list, you can send plant material for the DNA multiscan list. But with the DNA multiscan test, we will screen only for the fungi and bacteria on the list. But maybe the cause is another fungi or bacteria or a virus which is not on the DNA multiscan list. So if you don't know what's the cause, send it to a plant doctor. Maybe it's a virus, maybe it's a deficiency problem, or a fungi and bacteria not on the DNA multiscan list. So DNA multiscan is only for screening the fungi and bacteria on the list. So, at the end, where can I find microorganisms? I showed you a few examples. They can be on every place in the greenhouse. So check the right places. So make a list of possible places where pathogens can hide. and send a sample of this place. And choose also the right analysis method, also very important. And we can help you with this. More information about our analysis, you can visit our website. There is a specific website also for sending your samples. There are also sampling videos present on this website, and you can see them over there. Thanks for listening. Any questions, please do not hesitate to contact us. We will help you to find the right analysis for you and for your crop.